Nipro's Genoscholar series are diagnostic products for tuberculosis drug resistance. Nipro's Genoscholar detect genetic mutations of Mycobacterium tuberculosis, which are the cause of drug resistance to anti-tuberculosis drugs by the line probe assay. This line probe assay system consists of two steps, which are amplification step and detection step. First, prepare the master mix. Amplification solution and polymerase solution are mixed and diluted with distilled water to adjust the concentration of the amplification solution to 30 microliters test and that of the polymerase solution to 1 microliter test, respectively. The master mix is centrifuged for a few seconds before dispensing the single reaction mixture into each single PCR tube. The next step is addition of extracted DNA to the master mix. The extracted DNA is added to the master mix in a room special for this job using dedicated equipment. Because of a higher contamination risk, the master mix containing the extracted DNA in one tube should be dispensed into all sample tubes using a different tip for each sample. A negative control sample is also added to each batch of sample tubes. Before loading the samples into the thermal cycler, the tube should be centrifuged for a few seconds. Put the tubes in the thermal cycler, select the program, and press the button to start the reaction. The final PCR product consists of millions of copies of the target DNA. The next is reagent preparation. 5 kinds of reagents are prepared before used in this detection step. Bottle 1 is a hybridization solution. It should be kept warm before use. Bottle 2 is a rinse solution. This is prepared from the concentrated rinse solution by diluting 5 times with purified water. Bottle 3 is a conjugate solution. The solution is prepared by diluting the concentrated conjugate with a volume ratio of 1 to 100. Bottle 4 is purified water. And bottle 5 is a substrate solution, which is prepared by diluting the concentrated substrate solution with a substrate buffer with a volume ratio of 1 to 100. These reagents are placed in the multi-blot machine. To the hybridization solution bottle, insert the tube number 1. To the rinse solution bottle, the tube number 2. To the conjugate solution, the tube number 3. To the purified water bottle, the tube number 4. And the substrate solution, tube number 5. Tubes number 6 and number 7 are drain tubes to the waste bottle. Put the test strips into a multi-blot tray using tweezers. Pipette 10 microliters of the denaturization solution into the upper corner of each cell. Pipette 10 microliters of the PCR amplicon also into the upper corner of each cell. Open the front cover and unfasten the tray holder of the multi-blot. Place the tray on the heat block of the machine and fix the tray with a holder. Close the front cover. Select the program through the operation panel. Following the indication on the display, run the program. After the program is completed, open the front cover to take the tray out the machine. Each strip shows its own color development, depending on its genotype. The strips in cell 1 show the wild-type genotype. That means these are drug-sensitive. On the other hand, other 2 strips show mutant genotypes. These are drug-resistant. Nipro's Genoscholar series visualizes genotypes of the drug resistance gene of Mycobacterium tuberculosis and provides results within 1 day. Genotype is important information in drug selection.